So I'm Bob Handsaker. I'm going to get started so we don't run out of time, and so we can maybe have some time to talk and have questions as we go along or at the end. I'm in Steve McCarroll's lab at Harvard Medical School, and I'm also based here at the Broad in the Stanley Center and in the program for medical and population genetics, and I'm going to be talking about structural variation today and copy number variation. So this is a picture of a human genome. And the picture is created by staining the metaphase chromosomes. It's a technique that's been around for a long time. And if any of you are awake at 8:30 or observant, if you want to look and tell me if there's anything special about this genome, just go ahead and shout it out. I know some of you probably have seen this at the beginning of this presentation before, but yeah. So there's actually an extra copy of chromosome 21 down here. This genome is from somebody who has Down syndrome. And using techniques like the chromosome banding, we've been able to see things like Down syndrome for a long time. And here's another example. We could do the quiz here, but there's little arrows to help you out. If you look around, you can see there's lots of extra chromosomes and broken chromosomes in this, and this is because it's a tumor genome, right? This is something very common in cancer, and something you can also see at this kind of whole chromosome scale. And here's yet another example, maybe a little trickier, but it's a very famous example of one of an early discovery that was made. There's a recurrent translocation that happens in certain kinds of leukemia, especially CML, and it's a translocation between chromosome 22 and chromosome 9 that turns on a gene, and it's called the Philadelphia chromosome because it was one of the early discoveries about this powerful sort of recurrent translocation that drives cancer. And this is, again, something we could see with this. This was reported like around 1960. And so even with the technologies of the day, we could see things that had medical relevance to cancer at this really large scale. But to some extent, interrogating structural variation was hampered by the technologies of the time, right? So as the technologies advanced, we began to learn more and more about structural variation. And around 2000, an important technique that was developed was comparative genomic hybridization. And this is where you take two DNA samples and you label them so that they will fluoresce with different colors, and then you have them compete for hybridization. In the early cases, you had them hybridized to a genome that you had fixed. That's kind of like your reference genome. And then you look for just the abundance of the light. If there's more red light, and that means there's some green DNA missing, or there's extra red DNA that's present, red labeled DNA, and vice versa. And so you can begin to call gains and losses like this. And within a few years, we had figured out how to scale this up by putting it on microarrays. So you can put the probes on microarrays, and then you can begin to use smaller probes. And doing this, you can begin to ascertain copy number variation down to below megabase scale. And as this technology was developed, we began to realize that copy number variation happens not just in aberrant cases like Down syndrome or in cancer, but actually that there's abundant copy number variation in the healthy population, right? There's many segments of the genome and genes where we all have different copies, or most of us, if you look, will have different numbers of copies. In array CGH, even though it's a technique that's been around for a while, is for many years been and the gold standard in how we do certain assays for copy number variation, especially in the clinic, although it's being replaced more and more today with sequencing-based assays. So this was also the era when GWAS arrays were beginning to be used at large scale. And people realized that you could also use the intensity signal from the GWAS arrays to ascertain copy number variation. All those SNP probes have an intensity signal, and you're measuring the difference between the A and B signals. But if there's a copy number variation, if there's a deletion or a duplication, you'll get a shift in the intensity of the signal, and you can use that to call copy number variation. And with arrays, people still call copy number variance from arrays today, and you can get down to a resolution, below 100 kb, maybe down to 50 kb, or in some cases below that. It depends a little bit on how many probes you have. And actually, some probes were designed, some arrays were designed with probes specifically to detect copy number variation under common known CNVs and not just rely on the SNP probes. And Steve McCarroll and colleagues at the Broad did a lot of work on both the algorithms for calling from the array data, as did many other groups, and also on the design of some of the early arrays. So this was also the era when short-read sequencing began to become a really cost-effective technology for looking at the genome, and of course it's continued to drop in price. When you have short-read sequencing, then you have sort of this problem of trying to ascertain these very large structural variations from very short reads. So the scale of the read doesn't really match the scale of the variant you're trying to detect. So it means you have to use some tricks, I don't know if I want to call them tricks, but you have to look at the signals you get from the short reads to try to infer the larger copy number variants. And there's a few themes that people have traditionally used to analyze short-read sequencing data. So most of the short-read sequencing data is paired-end sequencing. So you have you fragment the DNA into small fragments, maybe 300 to 500 base pairs, and then you sequence from both ends of it. So you get two reads that are actually matched and should be pointing in a certain direction based on the library preparation technique. And then when you align those reads back to the genome, like you would to call SNPs, if there's no copy number variation, the reads will align 300 or 500 base pairs apart. But if there's copy number variation, for example, there's a deletion or there's a duplication in the genome, that pattern will be disrupted. So you can look at whether or not you have reads that have an incorrect orientation or they're spaced too far apart, and you can infer that there was a structural change going on in the genome you sequenced compared to the reference that you aligned the reads to. Another thing you can do with short-read sequencing data is you can just look at the abundance of the reads. In the same way you look at the intensity signal from a SNP array or from array CGH, you can just look at how many reads you get in a certain portion of the genome. If there's more DNA, you're going to sample it more times, you're going to get more reads, and so you can detect deletions and duplications that way. And if you have long reads, today we have usually 151 base pair reads, so you might get lucky and the read will land right on top of a junction that's in your genome but not in the reference. So if there's a deletion, for example, you'll have a new junction that's not in the reference genome, and you'll see reads that span right across it. And this works well in cases where you can uniquely align the reads. One of the difficulties with split read analysis is that a lot of copy number variation happens in repetitive parts of the genome where it's difficult to align the reads. So it's not as easy as just aligning all the split reads, but there's a large class of deletions and duplications you can find just from that kind of analysis. So sequencing comes in a couple different forms. We can do exome sequencing, for example, and early on exome sequencing was much cheaper than whole genome sequencing, so there was a lot of exome sequencing done. And you can call copy number variation from exome sequencing. Mostly you end up using that depth of coverage signal because you're only, in exome sequencing, you're using usually an array to capture just the DNA around the exome, so you're enriching for the exons. And so it's less likely that you would capture a breakpoint or that you would get read pairs that were spanning a structural variation. So mostly in exome sequencing, you look at the depth of coverage and you try to use that to infer copy number. It's a little bit technically challenging because the capture process actually introduces a lot of variation into the DNA in abundance. Some capture probes work better than others, so you get this technical noise that you have to deconvolve. But there are a number of methods that have been developed, including there's a sort of a new method in the GATK to do this. In general, these methods tend to the scale of copy number variation you can detect generally depends on how many different targets. So if a target is one exon, if a copy number variant spans 2 or 3 exons, you usually have pretty good ability to detect it. It's much harder to detect things that only hit 1 exon. And of course, if it doesn't hit an exon, you're out of luck. So from whole genome sequencing, we can also then look across the whole genome. We're not just limited to the exome. And a lot of methods have been developed to try to detect copy number variation, structural variation from whole genome sequencing. We began working on this problem in the early days of the 1,000 Genomes project with a lot of other groups. And we developed one of the early methods to do this called genome strip, which we still work on and extend today. Genome strip, I think one of the advances genome strip made was we showed that it was possible to get really high specificity and sensitivity out of the whole genome sequencing data, even with the low coverage sequencing that was done in the initial 1,000 Genomes project. And there were two fundamental things that we did that really turned up the sensitivity and specificity. The first thing was we combined multiple signals, right? In the early days, people would just look at read depth, or they would just look at read pairs or split reads, but we found that by combining the signals that this really improved performance, and that's become a pretty standard practice today. The second thing was that we looked across multiple people. So we're looking at a whole population of genomes together, and this was really useful for reducing technical noise and for using some of the statistical properties you would expect from a real segregating genetic variant to be able to separate good calls from bad calls. Another important theme I would say that emerged in the 1,000 Genomes project is that sometimes it's really useful to have specialized methods for specialized kinds of variants. So for example, there are mobile elements in the human genome. These are retrotransposons that are active in the human lineage. They're generally copy and paste retroelements, so that there's one copy in the genome and it will make a copy of itself somewhere else. And these are active in the human genome. You know, in all your genomes, there are ALUs that have jumped around and are existing in different places. Many of these are polymorphic. So the ALU jumps once, and then throughout human demographics, some people will have a new copy of the ALU and some people won't. So because these have very specific sequence signatures, and they're also very abundant, right? So ALUs comprise like 10% of the genome. So when you go to align an ALU read to the genome, it can go to many, many places, right? So this in some ways complicates the problem of detecting these polymorphic ALUs. So specialized methods that know about ALUs and L1s and their properties are much more effective at being able to look at these kinds of variants. And one of the popular ones is MELT by the University of Maryland. And this follows on really some of the initial work on this was done by Chip Stewart in the 1,000 Genomes project, and Chip is now at the Broad. Another technology that you can use to get at structural variation is these long insert jumping libraries. And the Talkowski Lab has been certainly one of the leaders and are very experienced in building these kinds of libraries. So the idea is, if you think about using read pairs to detect structural variation, looking for the rearrangements of the read pairs, you're limited by when the read pair actually lands over an interesting part of a structural variant. So if you take longer molecules, 3 kb or 5 kb, and sequence them, and you just sequence the end, you get read pairs, but now the read pairs are much more likely to hit a structural variant. So you're increasing your power to detect large structural variations by sequencing long molecules at the same depth as you would sequence shorter molecular fragments. So you can use this to find all sorts of structural rearrangements. It's a little bit of an art to create the libraries, and this is a little bit of an impediment to scaling this up, but it's a very powerful technique for getting at certain kinds of structural variation. So as we began to get these short reads in large populations in the 1,000 Genomes project, we began to see more of the spectrum of structural variation that exists in the genome. We were able to find lots of deletions and lots of mobile element insertions. In the early days, it was a little bit harder to find duplications and copy number variants, but we began to build this catalog of structural variation that's segregating in the human population. So then, as long read technologies like PacBio came along, right, this gives you a different way to look at structural variation and more power in many ways to look at certain kinds of structural variation. So PacBio sequencing, you get very long reads, 10 or 20 kb, although the error rate, the base error rate, tends to be higher than Illumina sequencing. But these reads can go through longer stretches of DNA. And what we learned from the PacBio reads, first and foremost, is that there's a lot of places in the human genome where there's significant rearrangement, duplications and inversions and small rearrangements, and these are stable. Right? They're stable in the population. Some people have the rearranged version of that chromosome, some people don't, and they're in healthy, regular individuals, and they are transmitted in the population. Also, people have used long reads, often in conjunction with short reads, to So you use long reads to make these sort of high quality personal genomes. And sometimes you use long reads and short reads together, because if you can use the long PacBio reads to kind of get a scaffold across the genome, and then the short Illumina reads, which have a lower error rate, to sort of fill in and correct the errors in the PacBio reads. And doing that, you can create these very high quality genome assemblies that are essentially personal genomes. The goal is to get an assembly of one person and to get actually two assemblies, one for each haplotype. And if you can do that, I call them these sort of immaculate genomes, right? Then you can just read off you can just take that genome and align it to the reference, for example, and read off differences to the reference. Or you can align two people's personal genomes and look at the differences between them, and you can kind of read off the structural variation. So there's been a lot of efforts to do this and create reference sets. Because it's costly to do all the long read sequencing. So far, these are in low numbers of people that we've been able to do this with, tens of people. And it's a challenge to get a completely finished genome assembly, end to end. Methods keep improving, but it's a powerful way to build catalogs of structural variation. And as we begin to have these personal genomes and these long read technologies, we can begin to look, in even greater resolution at the spectrum of structural variation that exists in the human population. And what we see is this trend where there's a lot of, so there's a lot of SNPs, like 3 million SNPs, right, in every person. And then as you begin to look at indels, there's less indels, like 300,000 or something. And then as you begin to look at slightly longer variants, and you get up into the structural variation range, the number of variants, decreases in this kind of curve. And if you compare this to the 1,000 genomes that I brought back as a small subfigure here, you can see that one of the differences in 1,000 genomes, you sort of see this curve going from long variation towards short variation, but then it sort of trails off. And that's not because that biologically it was trailing off, and we knew this at the time, right? It was that our ability to detect those smaller variants from the Illumina sequencing was challenging, and it's still challenging today. It's very challenging to get variants that are longer than a read and less than, say, a KB. Right? It's very difficult to ascertain those variants just from Illumina data. But those variants really exist, and we can see that they exist in PacBio data. And the only exception to this that you'll see in many of these plots is that the transposable elements, especially the ALUs and the full-length L1s, will always show up as these spikes, because these are polymorphic in the human genome, and there's a lot of them because they're created by this different mechanism. Another useful technology in some cases is optical mapping, and there are optical mapping techniques that date back to the 1990s with work from David Schwartz. A more modern version is BioNano. And what these have in common is you take really long molecules, like a couple hundred kb, and then in the case of BioNano, for example, you use a nicking enzyme to nick the DNA at a fixed restriction site, and then you can fluorescently label those sites. So you get this kind of beads on a string when you image it, and you can see these little fluorescent spots along the long molecule. And then you can take many of these long molecules, and you can use the patterns of the restriction sites to sort of line them up together and get this assembly across the genome. And doing this, you can get into really repetitive sequence. You can go into centromeric DNA with these really long 200 kb molecules, and you can see large-scale rearrangements and duplications and deletions. The downside, of course, is that you're only seeing the pattern of the restriction sites. You're not seeing base pair resolution, but you can get a really good high-level picture. Another technology that has become available more recently and has a lot of promise for looking at structural variation is linked read sequencing from 10x Genomics. And the idea behind 10x is that you take, again, long molecules, 100 kb, if you can get them into your library without breaking them. And then you use this bead-based method in library preparation. And what you do is for each long molecule, like 100 kb, you sequence it very lightly. So you maybe get 10 or 20 reads from that molecule, but every one of those reads is barcoded with the same barcode. And then it's relatively inexpensive because once you've prepared the library, you just run it through Illumina sequencing. And so you get out sort of a regular Illumina read, except it's got this barcode that tells you what molecule it came from. So you'll have 10 or 20 molecules with the same barcode. You know they all came from the same place on the genome. So you get long-range connectivity information from these kind of sparse reads. And then you can sequence them to a total depth of like 30x. So you can use this long-range barcode information in several ways to get at structural variation. One is you can just look whether or not the barcodes are next to their barcode buddies, right? Because they should be. They all came from the same molecule. And if you plot that, these plots are showing a loop, something called the loop browser from 10x that allows you to visualize where the barcodes landed when you align the reads back to the genome. You can see that if you just have a normal diploid place in the genome, that all the barcodes on the are close to each other. That's what the plot on the left is showing, that all the barcodes are near each other. But when you have a structural rearrangement, you'll get distinctive patterns of the barcodes not being where they should be. They're next to, the molecule doesn't look contiguous when you align it to the reference. So on the right, this is kind of a distinctive pattern you would get from an inversion if the person was homozygous for the inversion. But that's not the only thing you could do with this linked read data. A very, very clever idea that you can do with it is you can take the linked read data, and step one is you just call SNPs from it, right? Because it's Illumina data. So you call SNPs and you find all the heterozygous SNPs. Then you've got this long-range barcode information, and you can use that to phase the SNPs. And these linked reads are really, really good at doing long-read phasing using the barcodes. So now you've got two phase sets of heterozygous SNPs. So step three is you go back, and now you take all your barcodes and you assign them either to haplotype 1 or haplotype 2. And even the reads that didn't have a SNP in them, by the barcode, they're linked to haplotype 1 or haplotype 2. So now you've partitioned your reads into the two haplotypes. And if you could do that perfectly, you've got a tremendous increase in power, because now you can go in and use read depth, for example, or other techniques to look at copy number variation in each haplotype separately. So what this plot is showing at the bottom is how you can take the reads and separate them into haplotype 1 and haplotype 2 here, although there's a certain number of reads that couldn't be assigned to either haplotype. But you can, the top track is actually showing the depth of coverage with all the reads combined, and then the red and blue tracks are separating into haplotype 1 and haplotype 2, and you can see that haplotype 2 has a deletion, although it's a lot harder to see it in the data when it's not separated by haplotype. And the last thing, maybe not the last thing, but a third thing you could do with these reads is just assemble them. You have this nice long-range connectivity information from the barcode, and you have regular Illumina short reads. And so 10x has an assembler called Supernova that continues to increase in the quality of assemblies. You still have to do fairly deep sequencing to get a good 10x assembly out of Supernova. I don't know exactly what depth, so I don't want to say, but I think to get a good assembly you have to get a little bit deeper than standard 25 to 30x. But you can do this and then sort of use the immaculate genome approach to just read off the structural variation. So that's kind of like the interaction between the advance of technology and what we can see in terms of structural variation as the technology improves. So I'd like to switch gears a little bit and talk a little bit about what are some of the general things we've learned about structural variation. Yeah, Dennis.
You didn't talk about nanopore.
No, I didn't. And it's probably because I don't know so much about it. So, and I did want to stop and say at some point, of course, the holy grail of structural variation detection would be if you had a long read with a low error rate that was inexpensive, right? So when that sort of technology comes along, then in theory we can just sequence everybody's genome with that technology and read off the structural variation as well as everything else. So I haven't worked with some of these, the nanopore and the other new long read technologies like that. But yeah, I think that is the future, and a lot of the inferential stuff we're doing, when you can do that and then do it at a population scale, because the cost comes down enough, then I think structural variation can become a solved problem, at least in the detection part.
This was a good survey of this, but many of us are now poised between whole exomic sequencing and whole genome sequencing. And the question is when and how to do it. And the question is there something right around the corner here that's worth waiting for, or is now really good enough that when you go to whole genome sequencing, you can get the vast majority of what you want for information, recognizing that there's a curve of improving sensitivity. Where are we in relation to an asymptote? Or is there something ready that, in 6 months, you withheld that, in 6 months you'd have a much better way to do this?
So to my knowledge, there's not anything, 6 months away that's going to be a real game changer. I think the real game changer would be, the cheap, error-free long read technology, and I don't, as far as I know, that's not 6 months away, especially on the cost curve. We work a lot with whole genome sequencing in our lab, mostly because it's abundant, it's cheap, we can get it at population scale, and we're trying to look at things at population scale. If you're looking at an individual patient or something with a small set of samples, then you have more choices to spend a little bit more money. You could go get PacBio sequencing. You could use 10x. You could try nanopore and some of these other things. I don't know exactly how I would answer what I would do. I don't know actually the cost trade-offs of all of them. Yeah.
How important is the error-free in the long reads to observe structural variation? How error-free does it have to be, and how far away are we with PacBio, for example?
Yeah, that's a very good question. And I will say that I've been working with the 1,000 Genomes Project on our latest sort of follow-on effort to the 1,000 Genomes Project, where we've taken 3 trios and done really deep PacBio sequencing, mostly on the children, but also using the PacBio sequencing on the parents to help do the children. And it's still a challenge, right? It's still a challenge to get all the copy number variation with the PacBio sequencing. 1 base pair indels, extremely difficult to call, from PacBio alone, right? The error rate is just too high. And even some common copy number variation, the error rate's high enough that it's very difficult to distinguish the 2 copies. And some of the best PacBio people in the world have been working with that data and try to do a good job calling. And the algorithms continue to improve, but there's limitations. Compared to Illumina, you can see a ton more. But is it perfect? It's not quite perfect yet. All right, so some of the general properties of structural variation I wanted to talk about. This one I sort of mentioned before, but it's this idea that as we go from smaller variants to larger variants, we get this decrease in number, both in each individual and in the population as a whole. So there's less large CNVs than small ones, with certain exceptions. And when we think about structural variation, traditionally what we do is we sort of break it up into this menagerie of different kinds of events. So for example, we think about just deletions and duplications, right? Are people losing DNA, usually relative to the reference, or are they gaining DNA? And if you think of the reference as kind of a population norm, then maybe this has some genetic significance too. In general, we see that deletions and duplications, if you set aside like mobile elements, are generally roughly equal. There's tens or thousands of them or more in each person. And you do have to be a little careful when people talk about numbers, and numbers of deletions and duplications are detecting, because there's this relationship between size and the number of variants, right? If a study is using a technology where they can get slightly shorter variants, they're going to find many more because there are many more to find. So the number is a little bit hard to talk about depending on like where you make the cutoff. And even if you make a cutoff at 50 base pairs and say 50 base pairs and up is a structural variation or something like that, it's an arbitrary cutoff. There's a lot of variation at the 40 base pair size. So one of the things that we've learned about inversions and translocations is that we kind of, in a textbook way, kind of want to have this simple model that an inversion is like 1 piece of DNA and it's flipped around in place in somebody's genome. But that actually turns out to be very rare. Most of the time you find an inverted sequence, you find some other sort of structural change going on at the same time. Either it's an inverted duplication that's a little ways away, there's some copy number changes at the boundaries, there's a couple inversions going on together. So it's not like there's a mechanism that goes around and flips a lot of DNA. It's more of a local rearrangement in the area. And this is a more sort of common form of inversion or translocation that goes on. We talked a little bit already about mobile element insertions, and even though these are an insertional event, right? Biologically, it's a copy and paste into a new place in the genome. once there's a polymorphic ALU or other mobile element and it's circulating in the population, then it may or may not be captured in the reference genome. So if you're comparing to the reference, often what you'll see is what looks like a deletion of an ALU. And that's really an ALU that's inserted and now copied and now has been captured in the reference. So when you think about mobile elements, you always have to think about the biological mechanism versus the way we detect it. Novel sequence insertions tend to be, relatively less abundant. And what I'm talking about in terms of novel sequence insertions here is really, DNA that we know to be human, or maybe we didn't even know was human, and it suddenly shows up in somebody's genome, and maybe we knew it was human, but we didn't know where it went, and we suddenly can figure out where it goes in somebody's genome. This tends to be relatively less abundant than deletions and duplications. I think overall what we see is copying and rearrangement of existing human DNA, not like massive gene transfer from other organisms or something like that. Although we have, in some ways, it may be if that was happening, it would be a difficult kind of variation to detect, and especially across the whole population. So I'm going to talk in a little bit more depth about multi-allelic CNVs, mostly because that's what I've tended to work on in my career and my research. So I'm going to focus a little bit on those, and also because I think they're really interesting and really important. So the traditional model of copy number variation is that most of the time people have two copies, and occasionally, we'll lose, we'll lose a section of DNA, and maybe that'll segregate in the population. Maybe occasionally we'll get like a tandem duplication. So some people will have 3 copies of a certain stretch of DNA. But as we began to try to do genome-wide population scale looks at this, we found that there was a lot of loci in the human genome that just didn't seem to follow this pattern, right? It looked like there was much more variation in the copy number, or the patterns were really different in different people. People seemed to have very different structural alleles. And so we became really interested in focusing on this and asking some questions like, at these loci where the patterns aren't so clear, what is the actual genomic structures that we're looking at? And how is this structure varying in the human population? How many different forms does it exist in, and at what frequencies? And how does it relate to disease, right? Are these different structures actually driving disease, and are they important for human health? So we did some work on this using data from the 1,000 Genomes Project and trying to do a very thorough ascertainment of this kind of copy number variation across the whole genome using 1,000 people from the 1,000 Genomes Project. And we focused on these regions of multi-allelic copy number variation. So what these histograms are showing here is across 1,000 people, what are the different copy number states at certain loci in the genome? And so for example, at these loci, maybe most people have like 4 copies instead of 2, but a lot of people have 3 copies or 2 copies or 7 copies of a particular gene. And so there's a great deal of variation in the human population in terms of just how many copies of this gene each of us have. And when we did this study, we ascertained of these large CNVs, like 8,000 CNVs, of which the multi-allelic ones were only a small fraction, like 10 or 15%. But even though they were a small fraction, they were disproportionately found to contain genes. And moreover, when you think about like how the gene dosage differs between different people, and by gene dosage here we mean like how many copies of the gene each individual has, we found that on average, if you take 2 people from this room, you probably have different copy numbers of at least 75 genes, right, between any 2 people in this room. And the vast majority of that difference is driven at these sites of multi-allelic copy number variation. It's not driven by simple deletions and duplications. And when we looked at this, we also realized there was an effect of frequency, right? So if you look at the rare variants that have like less than a 5% allele, minor allele frequency in the population, we found that there was a small proportion that were deletions, and then it was about 50-50 between biallelic duplications and these more complex multi-allelic CNVs. But then when we looked at the common variants, greater than 5%, the fraction of deletions stayed about the same, but there were almost no biallelic duplications, right? They were all multi-allelic CNVs, right? And so what does this tell us? Well, the hypothesis is that in some sense, having a biallelic deletion is a stable state, right? You can lose a section of DNA as long as it's not essential for life in the human population, and it will persist and different people will inherit it. But biallelic duplications are not stable in the same way, right? If a duplication arises in the human population, and then that allele begins to go to high frequency, that allele tends to meet other single copy alleles, or then it begins to meet other 2 copy alleles. And when this happens, there becomes ample opportunity for incorrect crossover during meiosis. And as soon as you start to get the incorrect crossover, then the 2 copy allele reverts to a 1 copy allele, or it expands to a 3 copy allele. And we believe this is why we don't see, you see a few, but not a lot of biallelic duplications that go to high frequency without then generating more complex alleles in the population. So we talked about like this quantitative relationship between the number of variants and the size. But another important thing that we've learned is that the phenotypic impact of variants kind of goes in the opposite direction. And what do we mean by phenotypic impact here? So one thing we looked at when we looked at the multi-allelic copy number variation is we used some RNA sequencing data from GeoVetus to look at what happens to gene expression when the copy numbers of the genes are changing. And in plants, for example, some plants anyway, there's a mechanism where if the plant acquires an extra copy of a gene, it just gets turned off because it's not good to go messing with the abundance of genes in your genome and the level of expression. But it turns out, apparently, humans don't have such a mechanism. Because in almost all cases, when we found a gene that was copy number variable, and then we looked at the expression data from the RNA sequencing, there's a linear correlation between the level of expression and the level of genes. More copies of the gene, more expression, right? So these copy number variants are clearly biologically active in the sense that they're changing the gene expression. And in the 1,000 genome study, they took a look at this, and they found that when they looked at EQTLs in lymphoblastoid cell lines, they found that the EQTLs were enriched up to 50-fold for being caused by structural variation. And then in a more recent study in GTEx by Colby Chang and Ira Hall's lab, they did a similar analysis, and they found that up to 3 to 7% of the EQTLs are likely to be driven by structural variants, even though the structural variants were 0.1% of the variants they were actually testing to be EQTLs. So these large changes are having a disproportionate effect on genes, right? Because a large, large duplication or deletion can have a potent effect on a gene, compared to SNPs. So even though they're less abundant, they have big phenotypic effects. So another important question that our lab has looked at is how well we can impute structural variations from SNPs. And probably most people in the audience know this, but imputation is the idea that if you have a variant that you didn't measure, for example, a SNP that wasn't on your array, how well can you predict the state of that variant just from the nearby variants, usually SNPs, that you did measure? And we'd like to be able to do it with copy number variation too, right? To be able to predict the copy number state from nearby SNPs. And it turns out that the answer to how well we can do this is it's mixed. So these biallelic deletions that are relatively stable in the genome, right, they actually impute pretty well. They impute about as well as SNPs to a first approximation. But duplications and these multi-allelic copy number variation don't. And the more multi-allelic they are, right, the worse they tend to impute from SNPs. It just means that those copy number states are not very well correlated in many cases with the SNP background. So one quick example of this is a particular CNV at HPR, haptoglobin-related protein. And you can see there's a couple different allelic forms. There's a deletion allele, which is generally Asian-specific, and it's a little bit longer. But of a little bit more interest to us, there's this duplication allele, and actually many duplication alleles that sit right over the HPR gene. These tend to be Africa-specific. And the HPR gene is very interesting because it helps confer resistance to trypanosomiasis, which I managed to say pretty well that time. And HPR also binds to APOL1, which has exhibited Africa, which has Africa-specific genetic variation too, and it's been shown to be related both to sleeping sickness and to kidney disease. And in particular, the resistance to trypanosomiasis tends to be correlated with places in Africa where sleeping sickness is endemic. But one of the interesting things about this as a copy number variant is that there's these multiple duplication alleles, and it's kind of what we call this runaway copy number variant. So a certain fraction of the population is copy number 2, but when you have more copies, it tends not to be 3 or 4, you tend to have 6 or 7 copies. And so what does this look like if you think about trying to impute this variant? So this plot is showing the horizontal, each little horizontal line here is a haplotype, and this is showing 280 haplotypes from people of African ancestry. And the vertical banding is showing you the different SNPs that are kind of like a barcode for each haplotype. And the center of the plot is the HPR gene, and the branches that are going out are showing you how the LD is decaying as you move away from the HPR gene and the frequency in the population by how thick the bar is. And those little black ticks are showing you where the high copy alleles are clustering in terms of the background SNPs. What you can see is that the high copy alleles are generally on this one fairly long, fairly uniform SNP haplotype. But within that haplotype, all the different duplication alleles, which have different copies on them, are all together. And in fact, there aren't a lot of SNPs that can distinguish one from the other. So when you try to impute this, it's fairly easy, or you can be fairly accurate to tell if somebody has this allele that tends to have high copy number on it, but to tell which duplication allele they have, which copy number, is actually much harder from surrounding SNPs. Probably because those high copy haplotypes are mutating faster than the surrounding SNP background. So we know that structural variation has a lot of impacts in rare Mendelian disease, but I want to talk a little bit about the impact of structural variation on common disease. So in his postdoc, Steve McCarroll was instrumental in discovering a very early example of a common CNV having an effect on a common disease, which was this deletion polymorphism upstream of IRGM, which he showed altered the expression of IRGM because it interfered with the promoter, and was associated with Crohn's disease. And in autism, Jonathan Sabot did some amazing work where he showed that there was this strong association between rare copy number variation and autism, and not like a specific CNV, right, that caused autism, but he showed that there was this burden of rare CNVs, that if you have a lot of these rare CNVs, that tends to be associated with people who have autism. And studies that followed up on this, corroborated this, and they show that there's some loci that tend to have these recurrent CNVs that confer risk of autism, but then even in addition to these loci with the recurrent CNVs, there's still a burden of risk that comes from having this excess copy number variation. And I think today we still don't understand exactly how this phenomenon works, but we know there's a definite relationship between an excess of CNVs, both at these loci and in general, and risk of autism. And there's kind of a similar story in schizophrenia, is that we know that there's schizophrenia risk that comes from recurrent deletions and duplications at specific loci in the genome, and also there's a subtle additional risk for just genome-wide burden of large CNVs. And each of these CNVs individually is quite rare, but the penetrance is fairly high, although in total, these rare CNVs still explain just a small percentage of the total risk burden for schizophrenia. Probably the most extensive study on this, at least that I know of to date, was done by Christian Marshall and Daniel Hauergen here at the Broad, they looked at 41,000 people in the PGC, and they found that across multiple studies, they got an overall odds ratio for this CNV burden of 1.1 and a significant p-value. So that's kind of the story of CNVs and schizophrenia. Something that we've also taken a look at a lot in our lab is more complex CNVs, not just sort of these large rare deletions and duplications, but what about these complex and multi-allelic copper number variants? And one of the approach that's yielded some success for us is to combine several techniques. First, we take very careful copy number measurements at the locus, and we figure out where the segments are that are changing in copy number. And then we infer from this the haplotypes, and we use family-based or statistical phasing to try to figure out all the different structures that are segregating in the population. And then we analyze those for relation to phenotype. One of the first examples where we did this was at the MAP tau locus on chromosome 17, and this was work led by Linda Betker. On chromosome 17, there's this large megabase inversion that's actually fairly common in the human population. So I want to say it's like 15% of the population or something has this megabase in a different orientation than everybody else. We call them the H1 and the H2 haplotypes. But there's also a lot of copy number variation that goes on around this inversion, within it and outside of it. And so these diagrams here are showing you the different segments and how they've been, changing in copy number on the different structural haplotypes. And then if you look at the frequency of these haplotypes in the population, you see that there's two in particular that have gone to really high frequency in Europeans, although they're pretty much absent in the rest of the world. And Linda found that, interestingly, both of these haplotypes involve different fusion genes, different fusions that involve the cancel 1 gene. And because of the large inversion, right, these two haplotypes probably have difficulty recombining. So these two haplotypes with these fusion genes have gone to high frequency independent of each other at this locus on chromosome 17. And this locus has been associated with various phenotypes, including like fecundity in Icelandic women, which was studied by Decode. Another example is the amylase. The amylase locus is actually a family of amylase genes, and all of them are polymorphic and exist in different copy numbers. For amylase 1, for example, copy numbers in the population range from 2 up to as much as 20 in different individuals. A whole bunch of us in this room have different numbers of copies of the amylase gene. So Christina Usher has done a lot of work in exploring the amylase locus, and we were able to show, us and a lot of other groups, that some of the early reports on an association to BMI was probably just not right because some of the measurement and the analysis of the haplotypes didn't take into account the full range of the structural haplotypes of the amylase locus. Linda and Ronnie Salem were also did an analysis of the HP locus where they found that contrary to what had been believed, that the ancestral copy probably had 2 copies, and they found an association to cholesterol levels. And probably the most famous locus that we've worked on in the McCarroll Lab is C4, which is a gene that's implicated in schizophrenia risk. And if you can see the GWAS plot in the Manhattan plot in the middle here, in all of the schizophrenia GWAS studies, there's this one towering peak on chromosome 6 at the MHC, and because it's such a strong association, it was the first one, identified in schizophrenia. But fine mapping in the MHC is very, very challenging. But Ashwin Sekar was able to go in and fine map this, and what he found was that the signal fine mapped to the C4 gene. And C4 gene is one of these complicated genes. You can see on the upper left, there's all sorts of different structural forms. It has actually a couple different forms, C4a and C4b, with different biological activity. There's a long and a short form. But Ashwin was able to show that the risk localized to C4, and in particular, what dominates the risk is the number of copies of C4a that a particular individual has. And I'm going to spend just a minute on sort of a really important theme that Ashwin found and utilized in this work. And it's this idea that we've talked about how these multi-allelic CNVs are recurrent, and this makes them difficult to impute, right? Because you can have the same structural form existing on multiple different SNP backgrounds. And Ashwin found that was true at C4, right? On the left, we're seeing the same structural form on multiple different SNP haplotypes, right? Because there's enough mutation going on at this locus that the same form can become recurrent. So this is kind of a curse in the sense that it makes it difficult to impute these structural alleles. But it's also can be a blessing, because if you can show that the phenotype associates with the structural form independent of the SNP haplotype, right? This is strong evidence that it's probably the structural, something about the structural allele that's driving the phenotype. And that's exactly in the analysis Ashwin used here at C4. And so lastly, I want to spend a minute talking about somatic variation and somatic CNVs. There's been a lot of interest the last couple years in looking at clonal hematopoiesis and somatic variation that occurs in blood cells, right? In your bone marrow cells and your hemopoietic stem cells. And work from groups in and around Broad here, the Ebert Lab and Giulio Genovese, showed that these clones appear in the blood of many people, not everybody in the population, but they appear in many people, and they're strongly correlated with age. There's this sort of hockey stick plot on the right that shows that this clonal hematopoiesis increases with age, and it always kind of gives me the willies, this plot as I get older. But there's and then more recently there was these clonal variants can be both SNPs and indels and also copy number variants. You can find precancer states that you can detect in the blood, even pre-diagnosis. And more recently, a report last year showed that this clonal hematopoiesis, if you take out the ones that look like they're precancerous, it still confers risk of coronary heart disease, maybe twofold risk of coronary heart disease just in people who develop these clones. And this is kind of a precursor to introducing that Poru and Giulio, Poru Low, who's at Brigham and Women's and formerly in the Price Lab, have been this year working on a new, more sensitive statistical method for detecting these somatic CNVs, basically by combining the AB allele frequencies with very good long-range phasing from EAGLE. And they've applied this method to the UK Biobank, where they've ascertained thousands and thousands of these mosaic copy number variants in blood, down to a fraction as low as like 1%. And Poru gave a very nice talk about this at ASHG this fall. And so they've shown that there's this abundant copy number variation, both deletions and duplications, and this copy number neutral loss of heterozygosity. And that's when you have, so in most cases you have a maternal haplotype and a paternal haplotype at every place in your genome. But sometimes what happens is that you lose the paternal haplotype and you get two copies of the maternal haplotype instead. And so what that creates is that you'll have no heterozygosity over that because you have two copies of the maternal haplotype. And they find that this, as well as these mosaic deletions and duplications, are quite common as somatic events in the blood, and they found a lot of interesting biology behind this. So you can watch for this, which hopefully will be in press soon. So we've talked about structural variation, that there's fewer structural variants than SNPs, but there's generally more impact per variant. Maybe 3 to 7% of EQTLs are driven by structural variation, even though there's many fewer of them in each individual and in the population. In some cases, especially the multi-allelic CNVs, there seems to be evidence that they can have faster mutation rates, which makes them difficult to impute. They're not well tagged by nearby SNPs. In some cases, they can be easier to interpret because they have definite effects. Like if you duplicate a gene, the expression of the gene goes up. Right? And there's, of course, more we could do. New technologies, would help a lot, including these error-free long read technologies. And especially it would help with things we struggle with today, which is better ascertainment of these kind of mid-size variants from Illumina sequencing and maybe the cost of PacBio sequencing. So I'd like to thank a lot of people who gave me material for this and stories to share, and especially to thank Steve McCarroll, who's been my mentor for many years and taught me pretty much everything I know about genetics. Thank you very much.